tertiary structures of proteins where there is interaction between the side chain amino acids of the protein molecule. So a large number of amino acids constitute a particular protein, and the side chain interactions and the proper folding of the protein is what is defined by the tertiary structure of the protein, which relates to its function also. The higher order structure of the protein is the quaternary structure of proteins, where if the protein is made up of more than one polypeptide chain, how these two polypeptide chains are linking each other and how they are in association with each other to perform the overall function of the protein. So these four different categories of proteins are the higher ordered structures in proteins. Biologically important macromolecules, all the proteins are, and the molecular masses ranges from 10,000 to 6 lakhs in general. They are found on the plasma membrane, in the cytosol, localized to specific organelles in the cells, and 50 to 80% of the mammalian proteins are glycoproteins. So they contain covalently bound glycan structures, and the concentrations of this glycan structure varies from 0.5 to 50%. 0.5 is the least percentage of carbohydrate that can be identified in a glycoprotein, and 50% is the highest that can be identified. How do you detect these glycan structures in a glycoprotein? To distinguish it from a regular proteins can be detected by spectrophotometric methods or by colorimetric methods. But a glycoprotein, the glycan content can be easily detected by treating a glycoprotein with phenol sulfuric acid reagent, where in presence of acids, the sugars lose the water molecules and form furfurols. And these furfurols condense with phenol and form an yellow-colored complex, which is measured at 490 nanometers. Another important reagent that can be utilized for detection of carbohydrates in general is the anthrone reagent. So by these two methods, one can say that a protein is a glycoprotein. Next slide, please. Now, what are the functions of the glycan portions in proteins? The glycan portion in the proteins provides stability to the proteins, facilitates proper folding of the proteins to attain the appropriate biological function, and improperly folded glycoproteins lack biological function and are either degraded in the cells or secreted out of the cell. The glycan portion may serve as a recognition marker for interaction with other proteins. The cell surface glycan portion in proteins plays a very important role in recognition of other proteins and ligands. It is not always mandatory that the glycan portion is responsible for the interaction with specific ligands. In other words, a glycoprotein may be present with glycan structures, but the glycan portion is not involved in the specific interaction. The other parts of the protein may be involved in the interaction. Lastly, the glycan portion might aid in the entry of specific viruses into the cells, and protein glycosylation is a very important event and is one of the most fascinating field of biochemistry that has provided a large number of clues to understand a variety of diseases that affect mankind. Next slide. Now, just as we know that there are 20 different amino acids make up a wide variety of proteins, the number of carbohydrates that make different types of glycan structures is much less than 10. For example, you have carbohydrates as monosaccharides like the glucose, fructose, galactose, fucose, D-mannose, D-glucose, beta-D-glucose, beta-D-mannose. Next slide, please. N-acetyl glucosamine, N-acetyl galactosamine, and sialic acid. So these are the major carbohydrates that are present as constituent units in a wide variety of glycan structures. If we look at a glycoprotein, the glycoprotein has got glycan structure, and this glycan structure is not loosely adhering to the protein molecule, but it is part of a protein molecule and it is covalently linked to the protein molecule. What are the ways by which the glycan structure can be linked to the protein molecule? In this diagram, you see two different ways by which the glycan structure can be linked. One is called as the N-Glycosidic linkage, where the glycan structure is linked to the nitrogen atom of an asparagine residue in the polypeptide chain. And this glycan structure is one type of glycan structure, and here you find a different type of glycan structure. The second major type of linkages in the glycoproteins is the O-glycosylation, wherein the glycan structure is linked to the serine or threonine amino acid in a side chain. And where are these specifically linked? This requires also a specific sequence motif where this particular glycan structure can be covalently bound to the glycoprotein. So these N-glycosylated glycoproteins and O-glycosylated proteins can be on the same polypeptide chain, or they can be also on different polypeptide chains. So that means a polypeptide chain can be glycosylated either by O-glycosylation or by N-glycosylation at different places within the polypeptide chain. As we go along, we will see that roughly 2/3 of the glycosylation sites are occupied by the glycan structures in glycoproteins. Now, there are methods to how we can remove the glycan structure. Thereby, people have understood how the glycan structure is actually involved in the total processes. This removal can be both enzymatic and chemical methods. In the enzymatic cleavage, you find that the N-glycosylation, an enzyme called as PNGASF, can be used, which selectively breaks this linkage between the glycan structure and the amino acid, thereby releasing the entire glycan unit. Similarly, for the serine and threonine glycosylated or the O-glycosylated glycoproteins, the glycan component can be removed using a chemical agent called as trifluoromethane sulfonic acid. Next slide. Next, once the glycoproteins are synthesized in the ER, then they have to move to the Golgi apparatus, where the large amount of glycosylation processing happens within the cells. So this is the Golgi apparatus is the major site where there is different types of processing enzymes are present for the appropriate processing of the glycan structures. As we go along, we will see the different things. Next slide, please. Next one. Coming to the lysosomes, lysosomes are acidic organelles that are present in the eukaryotic cells. They host a large number of glycoprotein enzymes, which are called as acid hydrolases, and these represent nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, and phospholipases. So a large degree of macromolecules can be degraded by a variety of these glycoproteins that are present in the lysosomes. The acidic composition within the lysosomes is maintained by a proton pump, and it's always at pH 5 that these different glycosyl acid hydrolases exhibit their biological function. And every lysosome is covered through a membrane, which is very rich in further glycoproteins, different types of glycoproteins, and it protects the intracellularly, internally present lysosomal enzyme within the matrix area. Next, please.
Shiv Kumar sir, you can turn on your video, sir.
Turn off.
Turn on your video, sir. It's currently off. So the
Video is
attendees will be.
Oh, turn camera. No, it is on only. Video.
Okay, sir. Okay, so you can continue.
Yeah, video is on. Now, there are different pathways to the lysosomes. For example, within the eukaryotic cell, how the glycoproteins are reaching the lysosomes, or how are the macromolecules reaching the lysosomes. Three different pathways are known. One is called as the phagocytosis, where large molecules or large organisms like a bacterium can enter the cell by the process of phagocytosis, where the entire bacterium is engulfed into the cell after it adheres to the plasma membrane. And then in the cytosol of the cell, it forms a phagosome, and this phagosome is targeted towards lysosome for further degradation so that the bacteria cannot affect the cell in any way. The second type of internalization is through the receptor-mediated endocytosis, where there are selected receptors on the cell membrane, and these can bind to small molecular weight components like the ligands, or they can also bind to large protein molecules, which can be either normal proteins or glycoproteins, and it can also bind to several viruses, and then this can be internalized by the forming a receptor ligand complex on the cell surface, and this receptor ligand is moving as in a vesicular structure into the cell, where it is converted into an early endosome, and then these early endosomes fuse to form a late endosome, and this late endosome ultimately fuses with lysosome, and the entire ligand that has been internalized is further degraded by the wide variety of enzymatic actions. In addition, you have the autophagy within the cell, where organelles like the mitochondria can be further degraded within the cell by through the process of autophagosome formation, and this degradation is again taking place through the lysosomal enzymes that are present. Next slide, please. One back. Now, the different oligosaccharides that are present in the uh, glycoproteins are N-linked oligosaccharides, because a large number of glycoproteins in the cells are comprising of N-linked oligosaccharides. And this oligosaccharide, can click once more. So yes, there are three distinct types of N-linked oligosaccharides called as the mannose-rich type or high mannose type oligosaccharides. Then you have a complex type of oligosaccharides, and you have hybrid type of oligosaccharides whose properties are in between mannose-rich type and the complex type. In the mannose-rich type oligosaccharides, you find that the glycan structure is largely composed of mannose residues, whereas in the complex type of oligosaccharides, there is trimming of these mannose residues, some of them, and you have only three mannose residues to which there are additional sugar molecules that are attached. And this selective addition of these sugar molecules takes place in the Golgi apparatus, as we see in the subsequent slides. And each step, there is an addition of a different sugar molecule. So thus there is a antennary structures that are developed within the core oligosaccharide based to the attachment to the protein molecule. So all this is processed glycan structure, which is necessary for the protein's function. And in the complex type of oligosaccharides, in addition to these sugars listed here, you also find selectively the sugar fucose. But in the hybrid type of sugars, you normally don't find fucose, but you find galactose and sialic acid as a single antennary structure, whereas you have a biantennary structure in case of complex oligosaccharides. Next slide, please. Next. Now, the different types of glycan structures that can be found in living organisms are depicted in this cartoon here. You find these are the different types of sugars: mannose, N-acetyl glucosamine, sialic acid, galactose, xylose, and fucose. And yeast has the high mannose type of oligosaccharides. The insect cells have mannose, N-acetyl glucosamine, and fucose. The animal cells, different cell types have different types of glycan structures. As you see here, there is a variety of glycan decorated on this core oligosaccharide. And in the plant systems, again, you find high mannose type oligosaccharides, you find polypeptide type oligosaccharides, which also contain fucose as one of the sugars. In the humans, again, you find a wide variety of glycan structures similar to what you find in other animal species based on the type of tissue and based on the location of the protein, either to a particular cellular organelle or on the plasma membrane. There is also variation in the composition of the sugar. Next slide, please. Now, when we consider the biosynthesis of proteins, we know that it is through the translation of a mRNA. The protein is generated by addition of amino acids at a specific sequence, specific fashion, and you generate the polypeptide chain. On the contrary, the glycan structure, however, is synthesized as a precursor molecule or as a pre-assembled molecule primarily by specific enzymatic mechanisms. In other words, the carbohydrate chains in glycoproteins are enzymatically generated and are covalently linked to the protein. Therefore, the quantities of the available processing enzymes are generally not sufficient to ensure the synthesis of uniform products. So this results in what is called as microheterogeneity. The glycoproteins have thus variable carbohydrate composition. A glycoprotein found in a liver cell, the same glycoprotein, if it is found in the muscle cell, it may have a slightly varied glycan structure or glycan composition, despite the fact that it may be performing the same function, because the levels of the enzymes which are involved in generating the glycan structures vary from cell type to cell type. And the glycosylation reactions occur in the lumen of the endoplasmic reticulum, in the lumina of the cis, medial, and the trans-Golgi vesicles. Together, this will generate finally the different types of glycan structures. For N-Glycosylation, a 14 carbohydrate glycan structure is generated enzymatically, which is co-translationally added to the polypeptide chain at a specific asparagine residue site. Further processing occurs in the Golgi apparatus. So we are today dealing mostly with the N-glycosylation pattern, but also I'll tell you briefly about the O-glycosylation. The O-glycosylation, where the sugar residues are added to the serine and threonine amino acid residues in the polypeptide chain. And a typical glycosylation reaction, you find that you have a protein to where there is serine or threonine residue, and you have a nucleoside diphosphate sugar, the NDP sugar as it is called, which has the sugar component attached to the nucleoside diphosphate. And then an enzyme glycosyl transferase facilitates the transfer of the glycan, the sugar moiety from the NDP sugar to the protein molecule. Therefore, there are a large number of nucleoside diphosphate sugars. And as early as in 1960s, late 1960s, Leloir has discovered that several types of nucleoside diphosphate sugars can be formed, wherein sugar residues can be linked to the nucleoside diphosphate sugars. For example, here UDP is generated from UTP and glucose, and ADP is generated from ATP and glucose. Similarly, the CDP can also be bound to other sugars, and GDP can be bound to mannose sugar. So different types of nucleoside diphosphate sugars are involved in the glycoprotein biosynthesis. Here you find different examples. For example, UDP can be bound to N-acetyl glucosamine, N-acetyl galactosamine, galactose, glucose, sialic acid to some extent, and also glucuronic acid and xylose. So when these sugars are linked to the nucleoside diphosphate sugars, they are also termed as the activated sugars. It is in this form that they can be transferred to specific acceptor molecules. On the other hand, for the sugars fucose and mannose, it is not UDP, but it is the GDP nucleoside diphosphate sugar. Similarly, CMP is what is used for the sialic acids. Next slide, please. Next. The biosynthesis of O-linked glycoproteins, this is a stepwise addition of sugar residues to the serine or threonine in the polypeptide chain. Initially, from UDP, N-acetyl galactosamine is added, and you generate a monosaccharide unit to the protein molecule. Now, this is extended by addition of the second sugar, galactose, and this extension can be modified further. To this galactose, again, a sialic acid residue can be added, or another N-acetyl glucosamine residue can be added. Depending on the nature of the glycoprotein, there is an addition of these specific sugars to these in the Golgi apparatus. This is all O-linked glycosylation, where the glycan portion is linked to the serine or threonine residues. The next step, the next major biosynthesis of the glycoproteins is the N-glycosylations, which occur in 3 steps. First is the synthesis of a lipid-linked oligosaccharide. Second is transfer of the oligosaccharide N block onto asparagine residues of the nascent polypeptides. And 3 is the trimming and elongation of the oligosaccharides. Next slide, please. Now, an important molecule called as a dolichol phosphate that is present in the ER membrane is responsible for this glycosylation process to be initiated. So this is basically consisting of, next slide please, isoprene units, which are about 15 to 19 in number. And these isoprene units have at the terminal, this is a typical isoprene unit. Such units 15 to 19 constitute a single dolichol phosphate molecule. Next slide please. And this dolichol is nothing but it's a molecule membrane bound material that is synthesized starting from HMG-CoA, which is also a precursor for the biosynthesis of cholesterol on one hand and ubiquinone on the other hand. So through the farnesyl phosphate formation, most of the biochemistry students would have known what cholesterol is synthesized from the HMG-CoA through the reductase enzyme and farnesyl phosphate. So the same pathway also leads to the biosynthesis of dolichol phosphate, and this dolichol phosphate can combine with free glucose or with free mannose to form dolichol phosphomannose or dolichol phosphoglucose, which are very important. And the human dolichol, next slide. The human dolichol phosphate, next slide. The human dolichol is an isoprenoid consisting of 19 to 20 isoprenoid units and about 90 to 100 carbon atoms in total. So this is what can bind to free glucose or free mannose, thereby forming dolichol phosphoglucose or dolichol phosphomannose. In addition, next slide. Next. So a core oligosaccharide is actually synthesized that is linked to the dolichol phosphate, and this core oligosaccharide consists of 2 N-acetyl glucosamine residues, 9 mannose residues, and 3 glucose residues. So all the N-linked glycoproteins that are formed in the cells are always formed initially by attaching this 14 carbon unit sugar, which is also called as the core oligosaccharide. And this core oligosaccharide is generated by a variety of enzymes and is anchored to dolichol pyrophosphate in the ER membrane. How this is done, we'll see in the next 2-3 slides. Next, please. So the lipid-linked oligosaccharides are assembled sequentially at the cytosolic and luminal side of the endoplasmic reticulum membrane. Dolichol serves as a membrane-inserted lipid carrier of the lipid-linked oligosaccharides. Dolichol is also needed for the synthesis of dolichol phosphomannose and dolichol phosphoglucose. Full-length lipid-linked oligosaccharides are composed of dolichol, 2 phosphates, 2 N-acetyl glucosamine, 9 mannose, and 3 glucose residues, what I have shown in the previous slide. Next slide. Now, this is the ER membrane, and this curved unit is, each one is a dolichol unit, which is about 19 to 20 isoprenoid units, and with a phosphate group, dolichol phosphate. Now, this dolichol phosphate adds on N-acetyl glucosamine from UDP N-acetyl glucosamine and generates a phosphate and N-acetyl glucosamine, thus forming dolichol pyrophosphate attached with N-acetyl glucosamine. This is the first event that happens in the glycoprotein biosynthesis, where the core oligosaccharide is synthesized bound to the dolichol pyrophosphate. Once the N-acetyl glucosamine is formed, a second N-acetyl glucosamine is added on, and you get 2 N-acetyl glucosamines. After this is generated, in a sequence of 5 reactions, 5 mannose residues, each from 1 GDP mannose, are added sequentially to generate a specific linkage, and you get 5 mannose residues, and which are linked to the 2 N-acetyl glucosamine, which is anchored to the dolichol pyrophosphate. Now, this 7 carbon unit, once it is generated, is towards the cytosolic phase and is now flipped into the ER lumen side, and this mechanism facilitates further addition of sugars in the ER lumen, wherein additional mannose residues, 4 more residues are added from GDP mannose, and you get the 9 mannose residues. Subsequently, 3 UDP glucose residues are transferred to this mannose residue, and you add on 3 glucose units. So thus, this is the core oligosaccharide, which consists of 2 N-acetyl glucosamine, 9 mannose, and 3 glucose. And the N-acetyl glucosamine is linked to the pyrophosphate, and the pyrophosphate is part of the dolichol molecule. Now, this is the core oligosaccharide that is specifically added to the glycoproteins, where there is a specific sequence of asparagine, X, serine, or threonine. So this process is catalyzed by an enzyme called as oligosaccharide protein transferase. Therefore, the process of protein glycosylation is a co-translational process. So as the protein is getting folded, you get the addition of the glycan structure in all N-glycosylated proteins, you generate this 14-core oligosaccharide structure, which is transferred co-translationally to the glycoproteins. Yeah. This slide shows you the oligosaccharide transferase enzyme, which is located here, and this is the nitrogen atom in the molecule, which is bind asparagine residue, which is the nitrogen atom of asparagine, and to this nitrogen, this core oligosaccharide is attached. So 2-N-acetylglucosamine, 9 mannose, and 3 glucose residues is what is attached here. The enzyme oligosaccharide transferase catalyzes this specific reaction. So N-block, the dolichol pyrophosphate, is then recycled to perform the same reactions, and co or post-translationally, the oligosaccharide transferase complex facilitates this. And in the entire protein molecule, if there are 10 sites, about 2/3 of them are occupied by the glycan structures. Not all the glycosylation sites can be occupied because some of them may be folded within the polypeptide chain and may not be accessible for the addition of the glycan structure. The next important aspect about the glycoprotein biosynthesis is once this core oligosaccharide is generated, it has to be processed further. And this processing occurs in the Golgi apparatus, which is consisting of 3 independent units sequentially arranged: the cis Golgi, the medial Golgi, and the trans Golgi network. Next slide, please. Next. The processing of the glycan part in the Golgi apparatus is what's called as the sorting mechanism, where phosphorylation of the oligosaccharides happens for all the lysosomal glycoproteins. And in some glycoproteins, there is removal of mannose residue and addition of N-acetylglucosamine, addition of galactose, addition of sialic acid, and then further modification by sulphation of the tyrosine residues and the carbohydrates. And then they are sorted to the lysosome or to the plasma membrane. These are the 3 regions of the Golgi apparatus where the processing of the glycan structures happen. So for the first step of processing in the Golgi apparatus, what happens is that the 3 glucose residues are removed, and it is necessary to first add in the ER lumen these 3 glucose residues. Therefore, you have a core oligosaccharide generated, which can be accepted by a polypeptide that is growing to the oligosaccharide transferase. Subsequently, these glucose residues are not necessary. Therefore, they are trimmed by 2 independent enzymes, the glucosidase enzymes in the ER itself, and then they are removed so that you get only the mannose structures attached to the core oligosaccharide. Subsequently, an ER mannosidase also removes one more mannose sugar, therefore forming a 8 mannose residue. This is the precursor of the different types of glycoproteins which can undergo further processing. And what are the different types of processing mechanisms that occur? In the endoplasmic reticulum, trimming of the mannose 9 oligosaccharide. In the cis Golgi, trimming to mannose 5 and N-acetylglucosamine. In the medial Golgi, elongation, trimming to N-acetylglucosamine, mannose 3 and N-acetylglucosamine. Addition of bisecting N-acetylglucosamine and or fucose to core trisaccharide. Additional N-acetylglucosamine for further antennae. These are all the different types of glycan structures that can be generated by this sequence of reactions in the Golgi apparatus. Next slide. Next, please. So the high mannose oligosaccharides have the same structures as the intermediates which are formed during the process of complex oligosaccharides. Here you see that the mannose is removed by the mannosidase enzyme and you generate a 5 mannose and 2 N-acetylglucosamine structure. Next slide, please. Now this adds on in one reaction where an N-acetylglucosamine residue is added specifically to this mannose. Therefore, you generate this N-acetylglucosamine to this oligosaccharide. In the next step, by another mannosidase enzyme, 2 of these mannose residues are removed and you get 3 mannose residues and 1 N-acetylglucosamine, to which another N-acetyl glucosamine residue and a fucose is added, so that you have fucose residue added here and N-acetyl glucosamine to this structure, generating the complex type of oligosaccharide. Essentially, complex means that they have different types of sugars that are attached to the oligosaccharide unit. Next slide. Next. So there are other types of modifications that occur. So wherein there is addition of other sugar residues like the galactose residues and the N-acetyl glucose. This is additional, one more N-acetyl glucosamine is added here, and subsequently 3 galactose residues are added to the 3 N-acetyl glucosamine residues. So they are further decorated, and finally to these three sialic acid residues are added. So this is again a complex type of oligosaccharide consisting of fucose, mannose, N-acetyl glucosamine, galactose, and sialic acid. So the sialic acid is one of the major component in a large number of glycoproteins, and here some glycoproteins end up with only 2 sialic acids, some end up with 3, and some end up only with 1 sialic acid residue. This again depends on the levels of the different transferase enzymes that are involved in the overall biosynthetic processes. Next slide, please. In the hybrid type of oligosaccharides, the 5 mannose and 1 N-acetyl glucosamine residue now adds on 1 N-acetyl glucosamine to this mannose, and then you generate a hybrid type of hybrid type of oligosaccharide, which is different from the complex type of oligosaccharide. Here you find that in a single antenna structure, you find N-acetyl glucosamine, galactose, and sialic acid, as well as fucose. There are no biantennary structures in the hybrid type of oligosaccharides. Next slide. Next. So the carbohydrate chains of different types can undergo additional modifications such as sulphation, phosphorylation, or acetylation. Sulfate groups can be added to the hydroxyls in N-acetyl glucosamine or mannose. They may regulate the secretion of certain glycoproteins and protect them against proteolysis. Phosphate groups are added to hydroxyl groups of mannose residues in lysosomal enzymes. They act as chemical signals targeting proteins to lysosomes. The hydroxyl groups of sialic acids can be acetylated, thereby protecting the glycoprotein structure after acetylation, so that there is less susceptibility of the glycoprotein structure for proteolytic digestion. Next slide, please. Click once more. Now, for the lysosomal enzymes, basically, they also contain the high mannose oligosaccharides. Please go back to. So these glycoproteins are modified so that the mannose residues are now acquiring a phosphate group. And this phosphate group is essential to be generated on the lysosomal enzymes, which will determine that, yes, these are the lysosomal enzymes and these need to be transported and selectively going to the lysosomes in the cells after their biosynthesis. So two scientists independently in the early 1980s, Professor Kornfeld from USA, Professor von Figura from Germany, they worked on these lysosomal enzymes and the sorting receptors, and they identified a phosphotransferase enzyme which is responsible for adding the phosphate group onto the mannose residues in the lysosomal enzymes. Next slide, please. Next. Yeah. Here you find this is the lysosomal protein with the N-acetyl glucosamine residues and the mannose residues. And this mannose residue is now getting phosphorylated by a phosphotransferase enzyme, which adds on N-acetyl glucosamine phosphate to the mannose at the 6th hydroxyl position. So this generates this particular compound on the lysosomal enzyme. So all the 50 different lysosomal enzymes contain mannose oligosaccharide structures, and one of the mannose residues is modified to generate this phosphate group. The second reaction a phosphodiesterase enzyme removes the phosphate group and generates the phosphate group. And this phosphate is responsible for the selective transmission of the lysosomal enzymes to the lysosomes. Now, how these lysosomal enzymes are transported, we'll see in the subsequent slides. But in the absence of the phosphotransferase enzyme, what happens is the lysosomal enzymes, although they are synthesized, they do not carry the marker of the mannose-6-phosphate, and therefore they do not go to the lysosomes. They are secreted out of the cell, and therefore the lysosomes are lacking the lysosomal enzymes. And if the lysosomal enzymes are lacking, a lot of material, biological material accumulates in the cells, and then there is increase in the cell volume, and finally there is large vacuolar formation in the cells. This is what is called as eye cell disease, which appear like inclusion bodies in the cells. So eye cell disease is primarily because of the lack of the phosphotransferase enzyme, which was first detected in 1981, conclusively established in middle of 1980s. Next slide, please. Next. So in eye cell disease, which is detected also in infants initially, you find that there is a truncated growth and people do not normally survive beyond a particular time period. This is primarily because they lack the lysosomal enzymes, and therefore the multiple types of deficiencies occur, and there is resulting in death of the individual. Now, the transport of the lysosomal enzymes, essentially, in case of eye cell disease, there is no transport. So in the Golgi apparatus, you find that the processing occurs, but the processing does not yield the mannose-6-phosphate containing lysosomal enzymes because the phosphotransferase is absent. Therefore, the receptor cannot bind to this, and therefore the enzymes are secreted out of the cell, and they cannot get into the cell through the receptor, although it is present in the plasma membrane as well as in the Golgi apparatus, because they lack the recognition marker M6P. That's the reason why you get this disease. Yeah, next. You click a couple of times so that you get the points here. So the function of the receptor, which selectively targets the lysosomal enzymes, is to internalize the lysosomal enzymes that contain mannose-6-phosphate from the outside into the cell. For example, you have within the cell, you have the mannose-6-phosphate generated system when the phosphotransferase is present, and then the mannose-6-phosphate recognized by the receptor, and it transported to an endosomal compartment and finally to the lysosome. And from the outside, into the cell also, this receptor on the cell surface can bind the lysosomal enzyme or mannose-6-phosphate containing ligand. It can internalize them so that it can get into the cell. So what did we learn from this? We found that carbohydrates can serve as ligands for carbohydrate-specific receptors called lectins, and mannose-6-phosphate residues on lysosomal enzymes are recognized by receptors mediating the transport of lysosomal enzymes to lysosomes. Eye cell disease is a genetic disorder caused by the inability of the cells to generate mannose-6-phosphate marker. Yeah. So this different types of lysosomal enzymes can be targeted. Yes, these are the different types of lysosomal enzymes, and it is a typical lysosome structure which has a membrane surrounded which is highly glycosylated. And Christian D. Dewey in late 1950s first discovered lysosomes in cells in Italy, and then subsequently they have been shown to play crucial roles in a wide variety of cellular functions. And the size varies about less than 0.1 micrometer, and the volume, they occupy about 0.5% volume of the cytoplasm and contain largely acidic hydrolysis and several membrane glycoproteins. Now, the two receptors have been identified which are responsible for the specific targeting of the lysosomal enzymes to the lysosomes in cells. So these two receptors are called as cation-dependent receptor, or the MPR46, and the cation-independent receptor, or the MPR300. So both receptors are called as P-type lectins because they recognize the phosphate group on the mannose, on the lysosomal enzymes, bind to them, and then mediate their transport to the lysosomes within the cell. Both are transmembrane glycoproteins and they contain a short cytoplasmic tail, they contain a transmembrane domain, and they contain a extracellular domain. The difference between the 46 and 300 protein is that this has a single cassette structure in the amino terminal domain, whereas this has 15 repetitive cassette structures in the N-terminal domain. The function of all the 15 cassette structures is still not known, but what is known is domain 3 and domain 9 in the MPR300 protein is responsible for binding the lysosomal enzymes, whereas the domain 11 is known to bind specifically the IGF2. So the IGF2 is an insulin-like growth factor 2, which is in circulation, and this can be removed from circulation, and its levels are normally maintained so that it is not harmful. And this IGF2 is degraded in the lysosomes, and this receptor on the cell surface is involved in internalization of this circulating IGF2. The additional functions of this receptor are it also binds retinoic acid, human DNase 1, and thyroglobulin. However, the physiological significance of this binding is not clear. But intracellularly, both receptors mediate transport of the lysosomal enzymes. So they are coexisting, they are functionally similar, and their distribution is also similar in a wide variety of cells. So the combined effort of both receptors will facilitate proper lysosomal biogenesis pathway. And we have found in our studies extensively carried out on different lysosomal enzymes and the receptors that these are evolutionarily conserved proteins throughout the vertebrates. Therefore, the lysosomal biogenesis pathway involving a large number of glycoproteins is possibly same from fish to mammals. In addition, more recently, we discovered this in the Cnidarian hydra also, suggesting that it is a fairly very old phenomenon. The lysosomal biogenesis pathway is a very old phenomenon. Now, coming to the diseases that the lysosomal enzymes are involved in, why and how these diseases occur. Majority of the diseases, 1 in 7,700 live births, you get some sort of lysosomal disorder. There are about 50 different lysosomal disorders identified. Some examples are the Tay-Sachs disease, Gaucher's disease, Fabry's disease, Niemann-Pick's disease, Krebs disease, and so on. So these different diseases were initially discovered by two scientists by name Hickman and then Elizabeth Neufeld in US. So for example, the absence of two distinct enzymes, next slide please, absence of two distinct enzymes, hexosaminidase A and hexosaminidase B, will lead to Tay-Sachs disease and Sandhoff disease. So these are involved in removal of N-acetyl-glucosamine or N-acetyl-galactosamine sugar from different glycolipids and gangliosides in the neurological tissue. And absence of this will lead to accumulation of these particular glycan structures, thereby causing different types of neurological disorders. Next slide. So the several other known diseases like the Gaucher disease is known, which is an inherited metabolic disorder. Glucocerebrosidase is the enzyme that is deficient, and specifically the glycolipid glucocerebroside is accumulated in higher concentrations under these conditions. In Fabry disease, which is again a rare disorder, alpha-galactosidase A enzyme is absent. In mannosidosis, alpha-mannosidosis enzyme is absent. Therefore, there is accumulation of mannose containing glycan structures, and therefore there is this particular disease is prevalent. In all these disease conditions, the general therapy that is available to some extent, which is a very, very expensive therapy, is the enzyme replacement therapy. So what people do is recombinant human proteins are synthesized, and these are infused into the subjects which are suffering from this particular metabolic disorder. Therefore, they can be internalized into these cells. The lack of phosphotransferase enzyme leads to the absence of these particular enzymes. Therefore, these enzymes are now externally supplemented to these individuals. But these individuals contain the receptors on the cell surface, which can bind to the infused enzyme, and this enzyme can be taken up into the cells where it can be further processed and can be utilized for degrading various biological materials. The second and the more recent type of application-oriented work is on nanoparticle-based therapies, where you can use nanoparticle material. For example, you can use biologically degradable nanoparticles like the phosphorylated oligosaccharides, or you can use directly the lysosomal enzymes themselves as nanoparticles, which are much more efficient than the freely available lysosomal enzymes, and then use them to bind to specific receptors on cell surface. So here is a cartoon showing specifically how different types of lectins can be used as nanoparticles to bind onto the cell surface, and how they can be internalized into the cell. Thereby, since these are again protein molecules, they can be degraded in the cell to monomeric units, and therefore they are not toxic to the cells. Next slide, please. Next slide. Next slide. So there's a yeast secretes a phosphomanon called as orthophosphomanon, which has a highly branched polysaccharide with several mannose-6-phosphate residues. So this yeast phosphomanon is taken and it is hydrolyzed by acid, and then subsequently the acid hydrolysate is neutralized and then ethanol precipitated to generate two components. One is called as the phosphomanon core, which is a large molecular weight complex polysaccharide, and the second component is called as the pentamanosyl phosphate, which is a small molecular weight oligosaccharide. So both these contain mannose-6-phosphate residues and therefore can be used to purify the receptor proteins. And these can also bind the receptors on the cell surface. So what we have done is we have selectively made this phosphomanon core and pentamanosyl phosphate, and also some lysosomal enzymes, what we purified, we converted them into nanoparticles. Next slide. Next slide, please. So these nanoparticles are varying in size. Some of them are less than 100 nanometers in size, and these were characterized by transmission electron microscope in our end, as well as scanning electron microscope. And these nanoparticles basically are the enzyme nanoparticles. Beta hexosaminidase is an enzyme from lysosomal enzyme, and this is a beta galactosidase which is commercially procured. We made different types of lysosomal enzyme nanoparticles and checked whether these nanoparticles can bind the cell surface mannose phosphate receptor. The next slide shows indeed, yes, they can bind. And this receptor, next slide, this receptor on the cell surface, the MPR 300, after binding, can internalize the enzymes into the cells. And this internalization process is depicted here, where it can bind to and the primary purpose of doing this is to use the oligosaccharide molecules or oligosaccharide molecules tagged with specific drugs. Then we can use this receptor as a tool on the cell surface to internalize the nanoparticles into the cell. And these nanoparticles, being biologically functional, they will release inside the cell the drug, and the nanoparticles are non-harmful. They are not at all harmful because they can be biodegradable. So this nanoparticle-based approach for therapy of either lysosomal disorders or even other disorders like cancer and so on, these specific drugs can be tagged with the lysosomal enzyme nanoparticles or with the phosphorylated oligosaccharides nanoparticles and can be internalized through the receptor on the cell surface. So this is specifically in two different cell types. We have seen that there's a cell surface binding, and then once we increase the temperature to 37 degrees, there's an internalization of the externally bound ligand into the cell. This is a co-localization stating that it is indeed internalized into the cell. And this is detected specifically using an antibody to the receptor. And these are confocal microscopic images, which suggest that indeed the receptor on the cell surface is able to specifically bind to the nanoparticles that we have included. Next slide, please. And this is highly specific because we did these experiments also in presence of mannose-6-phosphate, and the mannose-6-phosphate prevents the binding of the nanoparticles to the receptor. You have high concentration of mannose-6-phosphate pre-incubated with the cells, so that saturates the receptor with the mannose-6-phosphate, thereby preventing the binding of the nanoparticles on the cell surface. That this explains that this binding phenomenon is highly specific. Next slide, please. Now, the different types of treatments, and these are the two major treatments that are known, and intravenous enzyme replacement therapy, bone marrow transplantation to slow disease progression, umbilical cord blood stem cell transplant to restore missing enzymes. These are all different types of therapies that are available, and the nanoparticle based approach could be an alternate therapy which could be effectively used in future. And the challenges that are for us towards understanding and treatment of the lysosomal disease are very many. Go back to the next slide. Previous slide. Early diagnosis, the delays in the treatment, lack of knowledge, and the expensive nature of treatment. So in our country, given the scenario of a large population and diversified populations in the country, we do not have a specific cataloging of how many patients are there with one particular lysosomal disorder, how we can treat them, how we can identify them, and so on. So a medical doctor, a pediatrician, because these are the ones which have to be detected very early during the infant stage itself by various genetic mechanisms and by different tests. There's one doctor, Madhulika Kabra from the All India Institute of Medical Sciences, Delhi. She has recently initiated a program where she's screening in Delhi different populations to identify different types of lysosomal diseases. So we're trying to establish links with her group, and also based on our expertise, we want to slowly prepare a catalog in the country where we can identify different types of lysosomal disorders and how we can categorize the different types of lysosomal disorders and then see how we can improve methods to treat these particular lysosomal disorders. I think this is the last slide. One more slide. Next one, please. Okay, to summarize what we have discussed today, largely with respect to glycoproteins and more preferentially towards lysosomal enzyme glycoproteins, which contain N-glycan structures, which are modified in the Golgi apparatus, and they are transported to lysosomes through a mannose-6-phosphate receptor dependent way intracellularly, and the same receptor on the cell surface can also internalize the ligands and target them to the lysosomes for degradation. There are some organisms like the Drosophila which lack both the receptors, but they contain the lysosomal enzymes. The question is, how is it that the lysosomal enzymes are transported to lysosomes in these organisms? It is possibly through a mannose-6-phosphate receptor independent pathway, which is still not understood and which needs to be dissected out. And there could be other proteins which are binding to the lysosomal enzymes and helping them to transport in the lower invertebrate species. But in all vertebrate species, starting from fish to mammals, this pathway of lysosomal biogenesis is highly conserved. And our more recent study on the Cnidarian Hydra vulgaris has unequivocally established that Hydra also contains lysosomal enzymes, and they also have a mannose-6-phosphate receptor protein. And Hydra we would like to use as a model system in future to understand more about these glycosylation processes and phenomenon, because this is totally unexplored area of research, and we would like to establish based on our expertise how the Hydra is in comparison with the human sample and how the lysosomal biogenesis takes place. And Hydra can be used as a good model system because the Hydra genome and the human genome is fairly closely related to one another. Thank you for your attention. I think I'll close here. This is the last slide, I think. Yeah.
Sir, there are few questions, sir, from the audience.
Yeah, where do I find them? In the
I will read out, sir. You please answer them.
ah,
One of faculty member has asked that, sir, can you please give an example of glycoprotein with
hmm.
0.5% and 50% glycan?
Okay. 0.5% is about there's a in the pea seeds in the legumes. Pea lectin has about 0.5% of glycan composition, and in another plant material that's the potatoes contain a lectin called as a potato lectin, which has about up to 50% of glycan structure. It's a highly hydroxyproline rich glycoprotein, which is also linked through serine and threonine hydroxyl groups, heavily glycosylated. As much as up to 50% of the protein is glycosylated. And even the mannose 6 phosphate receptor protein I was discussing about the MPR 46, the cation dependent receptor, is molecular mass is 46. 26 kilodalton is the protein molecular mass, and 20 kilodalton is the molecular mass of the glycan portion. It's highly glycosylated.
Yeah. Second question asked by the audience is, sir,
Yeah.
as you said, phosphomannan can be used as a therapy for lysosomal ASSO
Yeah.
diseases. Is this is an experimental stage, or any of these drugs are available for therapy?
No, not yet. In fact, the phosphomannan thing, actually we initiated this processes, and we there are a couple of ways by which one can do this. One has to also raise an antibody to the phosphomannan, which is fairly little difficult process. And the phosphomannan nanoparticles can be prepared. That's not an issue. But the question is, we need to actually establish clearly in different cell types, initially in vitro, and subsequently with animal model systems also we need to work. Then only we can go on for looking for any therapies. I would say it's another 3 to 4 years down the lane. We have just initiated the nanoparticle preparation. We are a little successful to show that indeed the phosphomannan nanoparticles can bind on the cell surface and can be internalized through the specifically through this receptor.
Sir, one more question.
Yeah.
For treating lysosomal storage diseases, enzyme or stem cell treatment is very expensive. Is there
Yes.
any inexpensive method?
So this is in any case this is the most expensive treatment, because even the enzyme replacement therapy is the one which is in use now. The other two are can be done. That's what is shown as examples, but they are not really. They are highly expensive. Even the enzyme replacement therapy is very expensive. There are no other cheap methods. We have to explore trying to develop newer methods which are relatively cheap. Possibly a nanoparticle based method would be a cheaper way.
Sir, one more question from the participants. Sir, what type of glycoprotein present on COVID-19? Because now we are in the pandemic stage. So maybe she is interested, she or he, interested to know what is the glycoprotein present on COVID-19.
You see, there's many glycoproteins. The spike protein is what is involved. They are saying, I don't want to really comment on that because I have not worked on it, nor did I have extensive literature survey on that. I am also listening regularly about these things, and because for this a new thing for everybody here, and I don't want to specifically comment on the. But thank you.
So I take this opportunity to thank Professor Siva Kumar from Department of Biochemistry and from Bhavan's College, sir. We are greatly indebted to you to spare your valuable time and then enlighten all the participants with your rich experience in glycoproteins. So the lecture is very informative and then very research oriented, sir. From the Department of Biochemistry and Bhavan's College, I express my immense thanks to you, sir, for sparing your valuable time and enriching everybody. Thank you very much, sir.
Thank you
So,
very much for this opportunity. I also enjoyed making presentation for this evening, and then I'm glad that a lot of people have registered and they were able to get some inputs from my side. Thank you so much.
so I inform
We
all the
will
participants